We were just looking at skeletal muscle in cross-section. That's what you see right here in 40x magnification. We can also look at skeletal muscle in longitudinal section. Now, once again, this particular slide doesn't show the staining that well, so I'm going to switch to a different slide. And actually, the staining is better, but you don't see what I want you to see. So here is our slide that says skeletal muscle and has in both in cross and longitudinal section. This is in longitudinal section. I'm going to get really close, 400x. Whoop. There. This is longitudinal section. You can actually see the band. See that light banding pattern? You can kind of see the striations of skeletal muscle here, but I have a different slide, one that is stained just for striated muscle, and I'm going to use that to show you striations and all the features of a muscle fiber. So the staining for this particular muscle is different so that the striations come out a lot better. So this is still longitudinal skeletal muscle, a longitudinal cut of skeletal muscle, 40x, 100x. You can see the individual fibers now going left to right, very, very long. And 400x, great. An individual muscle fiber, so it's sarcolemma, its plasma membrane, it can extend really, really long. I'm trying to trace it. It's not perfectly straight, but whoops. But this is the border between fibers, between cells. Really, really long fibers. They're that way because they started as myoblasts that then fused together to form a really, really long cell, which means if you have cells fusing together, these muscle fibers are going to have more than one nucleus. They are multinucleate. So we can see multiple nuclei. Here's the best example. Here's one nucleus. Here's another. Here's another. There's another. Many, many nuclei. With more nuclei, that means more DNA. With more DNA, that means more RNA. With more RNA, that means you can make more protein. And proteins are really, really important for skeletal muscle function. And you can see the protein, well, not individual proteins, but you can see the arrangement of proteins with these striations. Striations represent the arrangement of the proteins for contraction within muscle fibers. We see it in skeletal muscle, and we'll also see it in cardiac. So the darker lines are called anisotropic bands, but you can just call them A bands. The lighter lines are called I bands or isotropic bands. A bands are darker, I bands are lighter. And you have this repeating pattern over and over of dark and light bands. It's because of the repeating pattern of sarcomeres. And when I show you the sarcomere model, that'll make a lot more sense. Striations, once again, implies that there's a regular arrangement of proteins, and with this very regular arrangement, that can allow this muscle to produce, to generate a lot of force, comparative to, say, smooth muscle, which has similar proteins, but not the same kind of arrangements. So the amount of force generated by a smooth muscle won't be as great.